Page 12 of the Keystone Review Study Guide has a lot of questions about homeostasis and transport. I'm going to try to get it all on one video, but I might split it up into two. So the first question on page 12 asks us to identify and describe the cell structures involved in transport of materials into and out of and throughout a cell. So obviously we're talking about cell membrane type things here. So the first thing I'm going to go over with you is describe how the structure of the plasma membrane allows it to function as a regulatory structure and or protective barrier for a cell. Remember, you might see it written as a plasma membrane. It might be written as cell membrane. Be prepared to know what both of those are. And regulatory basically means that it regulates what enters and leaves the cell. So we know that that's the job of the cell membrane. Okay, a few nicknames that the cell membrane has. Sometimes it's called the phospholipid bilayer because it is made up of two layers of phospholipids, kind of like a sandwich. This is a phospholipid. It has a hydrophobic head. It hates water. Deconstructing the word phospholipid, you see lipid in there. Remember from Unit 2 that a lipid is a non-polar molecule like fats and oils and waxes. These are non-soluble in water, and they provide waterproof coverings for biological membranes. So the stuff we talked about in Unit 2 with lipids is coming back to haunt us here in Unit 3 when we're thinking about it in terms of homeostasis. The cell membrane, also known as the phospholipid bilayer, is also known as the fluid mosaic model. This is because it's not a rigid structure, it's a very flowy structure. And I like to think of it, and we talked about this in class, kind of like balloons that fill a pool. All these balloons are floating along the surface of the pool. They're touching each other, but they're not really stuck together too tightly. And if we threw a bucket of water on top of these balloons, the water would still be able to seep through or diffuse through. So that's kind of what this cell membrane is like. When we describe the cell membrane, we say that it is selectively permeable, meaning the cell membrane selects what can go in and what can go out. So it's a regulatory structure. It regulates what enters and leaves because it's selectively permeable. Other things to take a look at on the cell membrane, your two layers of phospholipids, obviously. This would be the outside of the cell. This would be the inside of the cell. There are some carbohydrate chains. There's some cholesterol embedded in there. They are involved in flexibility and communication with other cells. The big thing, too, to take notice of are these protein channels, also called transport proteins or integral proteins. The protein channels are involved in facilitated diffusion, which we're going to talk about in a little bit. There's also the ion channels, which are made of proteins, but these are in charge of your sodium and potassium pumps and your ion pumps. So that's basically the structure of the cell membrane, and it describes how it functions as a regulatory structure or a protective barrier for the cell. The next question we're going to look at is actually this part. Describe how membrane-bound cellular organelles facilitate the transport of materials within a cell. So the cell membrane is not the only membrane we talk about in biology class. We also talk about membrane-bound organelles on a pretty regular basis. So this picture here, and we went over this, I have this on the PowerPoint. You might be able to see it a little bit better in color. Let me go back to, okay. There it is in color. What you're looking at here on the left is the nucleus. Remember, inside of the nucleus is your DNA. There's your endoplasmic reticulum. And we can see that this is rough endoplasmic reticulum because it's studded with ribosomes, which are the site of protein synthesis. Over here, you have your Golgi apparatus, which is stacks of flattened sacs. And then here is your cell membrane. All right, so I'm going to go back to the picture where I took some notes for you. We're thinking about how these transport materials within a cell. Here you have your nucleus with your DNA trapped on the inside. Messenger RNA leaves the nucleus and goes and finds a ribosome. This is where protein synthesis takes place, or the making of proteins. Once that protein is constructed and travels through the ribosome, it's going to be packaged up in a little bit of a transport vesicle. This is just going to enable the protein to go from the endoplasmic reticulum over to the Golgi apparatus. Remember, the Golgi apparatus is the post office of the cell. Okay, this is where everything is packaged for transport, and it's added with some carbohydrates and fats to get it all packaged up in what's called a secretory vesicle. The secretory vesicle is then going to take this package of proteins, and either through exocytosis, it can expel it through the cell membrane, active transport, or it can send it somewhere else where it might be needed within the cell. But that's the basic idea. These vesicles are created by the endoplasmic reticulum and the Golgi apparatus to transport proteins around the cell where they need to be, or out of the cell if it needs to go to another cell to perform a job. I'm going to stop here, and I'm going to go on to the next part of page 12 in the next video. So hold on for page 12 part 2.